Hello, everyone. I'm Manu Saraswat. I'm a PhD student in the lab of Oliver Stegle in Heidelberg. Today we'll be talking about my PhD project, scDoRI, which is a deep learning framework to infer gene regulatory networks at a single cell resolution in a continuous manner. So in this audience, I don't need to explain what GRNs are, but what we need to know is that GRNs are regulators of cell identities. And changes in these GRNs are implicated across differentiation and development, as well as in diseases like cancer. Over the last years, single-cell multi-omic profiling assays have enabled studying of these cell identities and the GRNs at a much higher resolution than possible before. And typically, to analyze these data sets, and in particular, I'm going to be talking about single-cell multi-omic RNA attack data sets, the first line of analysis is to use latent variable models, which are essentially, let me use a laser pointer, which are a dimensionality reduction method, which brings down your higher dimensional data to a lower dimensional space. And then you can use that for clustering, identify continuous transitions like pseudotemporal ordering, etc. One example I'm showing here is of mouse gastrulation. And over here, you can see changes of blood progenitors to mature erythroids. And I'm going to come back to this example of mouse gastrulation later. Um, there's another like arsenal of tools which can infer gene regulatory networks from such multi-ohm data. Like when we infer these GRNs from multi-ohm data, there's like certain steps involved. Like first step is to find putative regulatory elements, and then you try to link those elements such as enhancers to gene, like in a correlation of accessibility and expression, like that sort of framework. Then you can look at what sequence features are present on those elements, the enhancers, then thereby making links between transcription factor to genes, get like one set of these links. And you can also use the correlation of TF expression and gene expression. And when I say correlation, I also mean machine learning model feature importance scores to further prune down those links. And one popular framework for the multi-ohm analysis of GRN is scenic plus. However, issue with this method and like a bunch of other methods is that they require either cluster level information or cell type level information, and that's the level at which they infer the GRN, or they infer a global GRN like for the entire data set, which means that they're not really utilizing the continuous nature of the data, the single cell, the power which is obtained by single cell data set. And the other issue is that, as I said, like they work in a stepwise manner, so you have to perform these stepwise analyses, they're not end to end. So the question we want to answer in our work is that can we bring together the best of both worlds? Can we combine latent variable models for dimensionality reduction with specialized GRN inference tools so that we have a framework which gives you a lower dimensional representation of each cell, where each latent dimension represents a GRN in itself. And having such a framework will really help us interpret such data sets in a very simple manner. And we want this model to be end-to-end so that we don't have to perform these steps like one by one. So to this end, we developed scDoRI, single-cell deep multi-omic regulatory inference. Uh, from like a very broad overview, it's an autoencoder-based framework, which takes the single-cell multi-ohm profile of chromatin accessibility and gene expression of a single cell or metacell as input. You feed it to an encoder, which is essentially a dimensionality reduction step, which brings down each cell to a lower dimension latent space. It's a topic modeling framework, so we call these latent dimensions as topics. We put some restrictions such that each cell is represented by a combination of topics, and these topics have to sum up to one. Now, the key novelty in this model is that instead of having fully connected neural networks as decoder, we use interpretable linear decoders to reconstruct the original profiles. Now, the advantage of using this linear decoder is that it allows us to interpret each topic as its own GRN. And when I say GRN, I mean a combination of transcription factors and their downstream genes. I will come back to it, like how it actually occurs in the next slides. But once we have this sort of model, we can use it for a bunch of downstream analysis. Like first thing you can do is you can take these topics, you can use it for clustering, identifying which topics are enriched in which cell types or trajectories. But you can also use them to identify which transcription factors are linked to each topic or latent dimension. And of course, since we are inferring enhancer GRNs for each topic, you also get the information that, okay, in each topic, this transcription factor is regulating this gene. So let's deep dive into this bit of the model, which is the interpretable linear decoder, which is really where the magic happens, like how like we get the interpretation of each topic. So it's a scary looking figure, and I will like go into it one by one, like what is going on here. So we want to reconstruct the accessibility profile of the cell. And to do so, we learn this matrix of topics times peaks. This essentially is a matrix of co-accessible peaks. So it tells you that, okay, in each topic, these are the set of peaks which are open at the same time. And you multiply it by the output from the encoder, which is the cells times topics matrix. And together you reconstruct the accessibility profile of each cell. In the second step, we want to reconstruct the gene expression profile. And to do so, we reuse the same matrix, like of co-accessible peaks. But at the same time, we learn another matrix of peaks times genes, which is telling you putative enhancer to gene links. Now to learn this matrix, like we put some constraint on distance as well. So if a peak is far away from the gene, it's likely to have less influence. If it's close by, it's likely to have more influence. But when you combine it together, you get the cells times genes matrix. So what essentially the model is doing here is that it identifies the set of enhancers, and if they are open in that particular topic, model says that okay, the gene is also likely to be expressed. Then in the third step, we learn this matrix of topics times TF expression, which combined with the cells times topics matrix gives you cells times TF expression. Now this is simply a denoised version of TF expression profile because we know that TF sometimes are lowly represented. So to overcome that sparsity, we learn the denoised version. And this matrix is like just a subset of the gene expression matrix. It's not something special, but just because they are transcription factors, we are learning this separately. And I'm saying the word step, like step one, step two, step three, but really you should keep in mind that all of this is happening simultaneously. It's not that like I'm doing it in a stepwise manner, like it's part of one huge decoder, and all these steps happen parallelly. And last and the final bit, which is the most interesting bit, is we learn this matrix of topics times genes. And it's not just one matrix, it's really composed of all the other things which we have learned before. So it tells you in each topic which genes are expressed, but this matrix is learned like through the GRNs. So we first learn what GRNs are active in each topic, and then we learn this matrix. So let's see how this is really constructed. So in the first step, we try to reconstruct topic-specific peak-to-gene links. So over here, I have already learned a matrix of peaks to genes, which tells me like, okay, for each gene, these are the set of peaks which could be acting as enhancers. However, not all of those peaks have to be open at the same time in all cell states. It might be that in one cell type, this particular peak is regulating the gene, other cell type, some other peak is regulating. So we take this matrix and we basically obtain like a filtered version of this matrix for each topic. And to obtain that filtered version, we use this co-accessible peaks matrix. Very simply saying, the model takes out the peaks which are not open in that topic and only keeps the peaks which are open, which could really regulate that gene in that particular topic. So you get these peaks times genes matrix in a topic-specific manner. Then we take these links and we multiply it with this transcription factor to peak links, which is in silico TF binding, in silico chip-seq matrix. It's essentially telling you like what is the probability of a transcription factor binding to a peak. And to compute this, we use like two lines of evidence. One is like the motif presence, which could be computed by Jasper scanning or using pre-trained TF binding models, which is what we are using. And the other line of evidence is that we use the correlation of TF expression with accessibility of the peak. So if across cell states, the expression of TF is linked to the accessibility of the peak, it's a high likelihood that the TF is actually binding there. And we get this matrix, and together, if you multiply these, you get a genes times TF matrix in a topic-specific manner. Now, these matrices are essentially the topic-specific enhancer GRN we are looking for. It tells you that, okay, in each topic, this transcription factor is linked to this gene, and it's a useful matrix to do downstream analysis. On the final step of this model, we use the denoised TF expression matrix to predict gene expression in a topic-specific manner. So we have the links that, okay, each gene is regulated by these TF, and we have the information of how much these TFs are expressed. You multiply them together and get this topics times genes matrix, which is fed here. So let's try to apply this to a real data set. And the data set we are using is a mouse castration data set of about 50,000 cells, 10x multi-ohm attack RNA from the same cell. So we apply scDoRI on this model and use the topic distribution of each cell to put them down on a UMAP and see if we can get like separation of cell types. And that is what we see, that we have the erythroid lineage here. This is more neural lineage. This is more mesodermal lineage. Then we can also look directly into the topics, like what they represent. And we see that there are certain topics which are enriched in a given cell type. For example, this topic over here is enriched in cardiomyocyte. This is particular for neural crest. And there's also a topic which very nicely captures the trajectory I was talking about before, like from blood progenitors to mature erythroids. You can see the topic is less active in progenitors, it becomes more active as you move along. There's also another topic which is like more shared across many different cell types, which are closely related mesodermal cell types, and it makes sense because they have shared gene expression program. There's a similar topic representing them. Now, one interesting example I want to highlight, and to highlight the importance of modeling these GRNs in a continuous manner, is this case of NMPs. So NMPs stand for neuromesodermal progenitors, and they can either differentiate to mesodermal lineage or like to neural lineages such as spinal cord. Now we wanted to see like what does the model learn as GRN for these NMPs, and we see that it learns two distinct GRNs. One GRN, which is active in NMPs, is also active in other mesodermal cell types. At the same time, there is this other GRN, which is active in certain NMPs, but it's highly active in spinal cord. So you see that very different, distinct GRNs are active in the same cell type. And if you were to use a cluster-based approach, some of these signals might be diluted. And since we are modeling it in a continuous manner, we get these very nice differences of these GRNs within the same cell type. And also a point to note here is that there are also intra-cell type heterogeneity, like some of the NMPs have higher enrichment for mesoderm, and like as you move along, like different away from the trajectory, the activation goes down, and like the opposite in the case of spinal cord. And since we have the interpretation of each of these GRNs and topics, so we can really take out this topic and see what is happening here. What are the TFs and genes here? So we take this TFs and genes topic specific matrix, and we do a gene set enrichment on the genes which are important here, and we see that they are enriched in nervous system development terms. And the transcription factors which are active here are SOX2 and PAX7, which are known neural transcription factors. And one last result I want to quickly highlight is that you can use these TF times genes matrix to also infer topic-specific TF activity. So this matrix tells you that each TF is regulating with genes, and you essentially just sum the values for each transcription factor, which tells you that what is the downstream role of each transcription factor, and you get this value for each TF in each topic, multiply it by the cell times topic matrix, and you can also get this information at a cell level. Over here, I'm showing a heat map of aggregated TF activities at a cell type level. And we can very nicely see that these TF activities can separate different lineages. And over here, I'm highlighting like GATA factors, which are coming up as important for erythroid lineage, and MEF2C and TBX20 and NKX25 important for cardiomyocytes. With that, I will conclude my talk. So just like a quick reminder, it's like a end-to-end framework for inferring GRNs at a continuous level. And since it's a deep learning model, it allows you to perform scalable inference, like it can work with thousands and millions of cells also. Yeah, happy to take questions. Thanks for listening.
Yeah, very nice talk. So actually, I have two questions. The first one is like, how do you decide the number of topics? So it's like, is there any way to specifically define them? For example, if we want to infer like cell type specific GRN, can we just fix this topic to be the cell types? Yeah, this is the first question.
Yeah, I mean, it's a good question. Like, we don't have a great answer to this. Like, as a heuristic, you can start off with, if you know the cell type information, you can roughly start with the same number of topics. And then you can try to vary up and down what gives you like better results. Like in this case of gastrulation, we had around 37 cell types, so I chose a number of about 50 topics. But then it brings you to a bigger question, like since this model is like unsupervised, you can ask a different question that you don't need any information about cell type. You can let the model decide what are the different set of GRNs which can explain the data well. And maybe that number is less than the existing number of cell types or more than that. So it's a very nice, interesting thing to see.
Okay, the second question is about the validation part. So since you infer like one GRN for one specific topic, and the topic is not specifically defined at the beginning when you obtain the data set, right? So is there any way that can quantitatively measure the accuracy of the GRN after you infer them?
Yeah, so validating the GRNs is always a very tricky aspect. So you're right that we don't refine already like what topic will represent, but some of the things we check is that, okay, if there is a topic coming up as important for a given cell type, and we already have some information about what TFs are active in that cell type, that gives us already some information. Also, the gene sets which are coming up for that particular cell type, do they make sense or not? And one another thing we are trying to do, which is similar to what Scenic Plus also did, was they tried to use like they created pseudo single cells from encode bulk data, and now you have information about a lot of chip-seq where TFs are binding and things like that. So you can further validate the model based on like that bulk data because there is a lot more evidence of which TFs bind to which genes and things like that. It's not perfect, but it's like a good start too.
So this kind of thing is like a whole data set based, right? It's not like topic based, right?
Sorry, what?
The chip-seq data after you obtain the TF binding. So
Yeah.
it's like a whole data based, right? It's like not specific topic based.
Like the in silico chip-seq I'm using here. This one you mean?
I mean, like when you said like validation,
A
you use bucket.
validation chip-seq is like encode like data from
Yeah.
these cell
So
lines.
yeah, it's like a data set. Like after the whole data set, you can obtain one like binding information, right?
Yes.
It's not like a topic specific.
I mean, so we do some mapping of topic to cell type. Let's put it that way.
Oh, okay, I see.
So like you train the model unsupervised way, but then you see like I also show in this example over here that there is a fixed topic, like this topic is only present in cardiomyocytes. So that also happens when you train it on encode data, and more so there because those cell lines are so distinct that there's one topic coming up for each of those. Then it's very easy to map the topic to cell type and then do further validation.
Thanks.